Welcome to my channel, Pure Biology. And today's topic is oxidative phosphorylation. What is the oxidative phosphorylation? This is the electron transport chain. Actually, during glycolysis and Krebs cycle, first of all, glycolysis and Krebs cycle I explained in my previous videos, and today I explain about the oxidative phosphorylation. So, during the process of glycolysis and Krebs cycle, ATP is formed, we know, but also the electrons remained within FADH2 and NADH, and these electrons released during the electron transport chain or the oxidative phosphorylation. Okay. Here, these electrons that are released from NADH and FADH2, these electrons added to, that means goes to oxygen, O2, and after that it helps to form H2, water. Okay. So actually the role of these electrons that remained within the NADH and FADH2 during the production of ATP, actually it helps, these electrons help to form water. Okay. It is added to oxygen and to form water. Here are the reactions. You can see NADH plus H plus and half O2 that helps to form water, and here that NAD plus form. And second step, FADH2 half O2 then form FAD and water. That means in two substances, in case of two substances, hydride, you can see the hydrogen ion that releases from that NADH and FADH2, and NADH changes to NAD plus, and FADH2 that changes to FAD. Okay. So this is the oxidative phosphorylation. Okay. So this oxidative phosphorylation occurs in inner mitochondrial spaces, and these are the enzymes that are involved for completion of this process. Okay. So this enzyme complex are known as complex 1, complex 2, complex 3 and complex 4. The first one is known as NADH coenzyme Q reductase, that is complex 1, or NADH dehydrogenase or NADH dehydrogenase. The second complex is succinate coenzyme Q reductase. Third one is coenzyme Q cytochrome C reductase or cytochrome BC1. This is the complex 3. And the last one, that is the cytochrome C oxidase. This is the complex 4. So these complex 1 to 4, these are all involved in the completion of this oxidative phosphorylation. And there are some phosphatic groups that are also involved. FNN, second one FAD and FES, third one heme, FES, and the fourth one heme, Cu plus. Okay. FNN, you know, this is known as the flavine mononucleotide. Flavine mononucleotide. Okay. Flavine mononucleotide. Okay. So, what are the enzymes? Complex 1, complex 2, complex 3 and complex 4. These are all the enzymes, and these are all the phosphatic groups that are involved in the completion of this reaction. That means electrons that helps oxygen and 2 hydrogen to form water. Okay. we will just learn very elaborately. Okay. Now we learn what is the role of this complex 1 and complex 2. Okay, in case of oxidative phosphorylation. So this complex 1 that constitute one FMA and one F6 to 7 APS. One FMA and 6 to 7 APS. So complex 1, it transfers electrons to the COQ, coenzyme Q, coenzyme Q. How the electrons are transferred from? That means complex 1, it transfers electron from NADH. It transfers electron from NADH. First of all, to the iron sulfur cluster. Okay. First of all, to iron sulfur cluster, ABS. Then from this ABS, it transfers the electrons to the COQ, coenzyme Q. Clear? So this is the role of complex 1 or the NADH reductase. Okay. The second one, that is the complex 2. Once again, here the protons, this complex 1, another role of this complex 1, that means the protons are pumped here. Protons are pumped here in the inner mitochondrial spaces. Okay. Second one, what is the role of the complex 2? First of all, complex 2, what involved in complex 2? FAD and FES. Actually, electrons here form during the formation of fumarate. Succinate to fumarate. Okay. Succinate to fumarate. The electrons are formed in that situation. These electrons, first of all, go towards FES, iron sulfur cluster. Okay. Then iron sulfur, from the iron sulfur cluster, these electrons go to the coenzyme Q. Okay, coenzyme Q. So, what is the role of complex 1? The transfer of electron from NADH to coenzyme Q, and complex 2, same role, the transfer of electrons occur here from FADH2 to the coenzyme Q. Clear? After that, we read or we learn about what is the role of the complex 3 and the complex 4. Sorry. And I think the role of the complex 1 and complex 2, is it clear? Okay. Next one. Next, what is the role of complex 3? Complex 3, within complex 3, there is heme and iron sulfur cluster, and in complex 4, there is also heme and copper. Okay. So, what is the role of complex 3? Actually, complex 1 and complex 2 both transfer the electron to coenzyme Q. COQ. The COQ that is present within the complex 3. Now the electrons, how transfer from the COQ? Okay. The COQ first of all transfer the electrons to the B-type cytochrome. B-type cytochrome. Okay. What is B-type? One is BH, another is BL. H for high affinity and L for low affinity. Okay. First of all, COQ transfers the electron to the D-type cytochrome. From here, the electrons are transferred to the C-type cytochrome. Okay? C-type cytochrome, that means cyto C1. Okay? And from cyto C1, the electrons are transferred to cytochrome C. Cytochrome C. Okay? And this is the oxidized form of cytochrome. This is the oxidized form of cytochrome. Oxidized form of cytochrome. What happens here? That means the complex 1, CON1 and CON2, complex 2, both transfer electron to COQ. Clear? Then from COQ, the electrons are transferred to the D-type cytochrome. That means BH and BL. BH, I think B's affinity is 566 and BL's affinity is 562. Here, from B-type cytochrome, the electrons are transferred to the C-type cytochrome. That is C1, actually C1. This is not the oxidized form of cytochrome. And from here, the electrons are transferred to the cytochrome C. This is the oxidized form of cytochrome. So, this is the role of the complex 3. And the last one, the complex 4. Here, the electrons, after entering into the oxidized form of cytochrome C, it enters into the complex 4. And what happens in complex 4? And what is the role of the Cu plus? This is very important. One electron, the electron, first go to the CuA and to cytochrome A. And second electron that goes to the CuB and to the cytochrome A3. Clear? Okay, the two electrons in that way transfers to the oxygen molecule, and generally complex 4 carries 1 electron for each copper ion. Okay, and here are 2 copper ions. I explained that. What is the copper A and copper B? So CuA, 1 electron goes to CuAA and to the CYTA, cytochrome A. And the second electron goes to CuB and to the cytochrome A3. Clear? And after that, these electrons enters to add with oxygen to form water. Clear? So, these are the roles of complex 4 and complex 3. And what is CuA and CuB? What is the role of, and what is, why we have written that CuA and CuB? CuA, actually CuA, it has linkage, or CuA, there one residue present, that is cysteine residue. And CuB, in there, histidine residue is present. Okay? And due to presence of these two different residues, we just name them CuA and CuB. Okay? These are the role of the complex 1, 2, 3 and 4. After that, we just read about what is cytochrome and what is coenzyme Q. So, continue after that sometime. Okay. Now we learn what is the role of coenzyme Q and cytochrome. That means not role. What is COQ and what are the form of each we find in the mitochondria or inner membrane spaces, and type of obviously cytochromes that are involved in this oxidative phosphorylation. COQ, that is coenzyme Q, its full form is ubiquinone. What is the name of this? Ubiquinone. Ubiquinone has three parts. One is fully oxidized form, another not part, that is forms. One is fully oxidized form, another is semi-ubiquinol form, and third one fully reduced form. The fully oxidized form known as ubiquinol A. Sorry, ubiquinol A, not ubiquinol. Okay, and Q, not Q. Okay. Second one, the semi-ubiquinol. Its name is ub-semiquinol Q. Clear? Ub-semiquinol, you can say. Fully reduced form, that is known as ubiquinol. Okay, ubiquinol. Its form is known as QH2. QH2. Okay. So three forms are there of ubiquinol. One is fully oxidized form, another is semi-ubiquinol form, and last one is fully reduced form. The fully oxidized form is known as ubiquinol Q. Semi-ubiquinol form is known as ub-semiquinol, and fully reduced form is known as ubiquinol, that means QH2. Like that, cytochrome has different types. One is B type, another is C and C1 type, another is A and A3 type. These are B type, C type and A type. Clear? The B type, that are known as B566 or B562. C type, there are C and C1, and A type, there are A and A3. Okay? So, today actually we know about what are the enzymes and what is the name of coenzyme Q and cytochrome, their types and the role of the enzymes, obviously. In the next video, I will discuss about the Q cycle, how the transportation of electron occurs and what are the inhibitors and how do we calculate the electron transport. So it's actually another thing, and I will discuss about these in my next video. Okay, thank you.